you ever get hungry? Are you interested in sea slugs? If you answered yes to either of those questions, stay tuned because this is the presentation for you. Hi, my name is Shane Ziemba, and today I'll be sharing with you my research on tracking down neuropeptide F in the brain of the nudibranch Bergia stephaniae. This work is done in collaboration with my mentors, Dr. Cheyenne Tate and Dr. Paul Katz at the University of Massachusetts Amherst. So what exactly is neuropeptide F? Well, neuropeptide F is a hormone responsible for increasing appetite and causing hunger. It's the invertebrate version of neuropeptide Y. So us humans have neuropeptide Y, but invertebrates like Bergia have neuropeptide F. A lot less research has been done with neuropeptide F when compared to neuropeptide Y. This is especially true for the receptors. Humans have 4 G-protein coupled receptors for neuropeptide Y. Neuropeptide F has never been looked at in Bergia, so it's completely unknown what the sequences are and how many receptors there are for neuropeptide F. So how was I able to find the neuropeptide F sequence for Bergia? Luckily enough, our lab sequenced out the complete transcriptome of Bergia. This is a complete list of all the messenger RNA that Bergia will produce. Using that, I was able to compare the transcriptome to other known neuropeptide F and neuropeptide Y sequences. Neuropeptide F is characterized by its C-terminal RX-RF amino acid sequence. You can see in the figure highlighted in green various NPF sequences, including Bergia's, that all contain the RX-RF motif. At the bottom is the human neuropeptide Y sequence, which contains an RX-RY. This is the difference between NPF and NPY, a tyrosine substituted for a phenylalanine. The same rationale for identifying neuropeptide F was also used for identifying neuropeptide F receptors. Sequences of known neuropeptide F receptors were compared to the transcriptome of Bergia to create a list of probable receptors. These probable Bergia NPF receptors were then compared again across the animal kingdom as well as to other G-protein coupled receptors, which is what NPF receptors are, to see how well they fit phylogenetically. After all that, I have identified 4 NPF receptors for Bergia. A hallmark of G-protein coupled receptors is that they have 7 transmembrane regions. I've highlighted the transmembrane regions in red. As you can see, the predicted receptors 3 and 4 are incomplete, which is a consequence of the sequencing technology. One of the great things about Bergia is that their brain has large and identifiable neurons. There are regions in the brain that contain specific neurotransmitters, peptides, and hormones. Pictured here is the brain of a Bergia that has been stained to show cells expressing neuropeptide F. This was done through a technique called in situ hybridization, which fluorescently labels mRNA containing a sequence of interest, in this case neuropeptide F. So you can see more of what I'm talking about with the brain on the left, which has been stained for all cell nuclei. We can clearly see that neuropeptide F is expressed in all major ganglia, the rhinophore, cerebral pleural, and pedal. The arrows point towards 4 neurons in the cerebral pleural that have the highest expression of neuropeptide F. This is because they have the most fluorescence. Next, we looked at the expression of one of the predicted NPF receptors, and we found that overall, more neurons express the NPF receptor than NPF itself. So now that we've localized the expression of both neuropeptide F and one of its receptors, we wanted to see what would happen to the expression as the Bergia got more hungry. We can clearly see that the Bergia that has been deprived of food for 1 week shows much higher expression of neuropeptide F as opposed to the Bergia that's been fed normally. In the circle, you can see that there is a large increase in expression in the cerebral pleural. This pattern wasn't there for the neuropeptide F receptor though. It seems that as Bergia becomes more deprived of food, the cells expressing the receptor change. Many of the identifiable neurons don't line up between the normally fed and 2-week food-deprived Bergia, meaning that the expression of the receptor for neuropeptide F is more variable as Bergia becomes more hungry. To summarize, Bergia does in fact have an NPF sequence, and I was able to identify four probable receptors for NPF. And NPF was shown to be found in all major ganglia, in cells of all shapes and sizes. Also, the receptor for neuropeptide F showed more expression than NPF itself. And as expected, neuropeptide F expression increased with food deprivation in specific regions of the brain, specifically the cerebral pleural. And lastly, the expression of the neuropeptide F receptor is more variable across the food deprivation levels. I would like to thank the CATS lab, as well as Jim and Maya at the light microscopy facility, for allowing this work to happen.